Good morning, students. So today we are going to discuss the second chapter or the second chapter's second part. We have already discussed the first part of the second chapter in the previous video lesson. So if you haven't gone through that lesson, please refer to that lesson first, then come to this one in order to get good understanding. So we are here discussing about sexual reproduction in flowering plants. So if we are studying basically about the mechanism by which the plants produce gametes, and these gametes fuse with each other to form a new plant. I am a biology teacher, Ankesh Singh. Hope you all know me very well. This is my mail ID and phone number. If you have any doubts regarding this chapter, regarding this subject, you can call me or reach me at this email address wherever is feasible to you. Let us move ahead and see our today's first topic, because we have discussed about the male part or the androecium in the last video session. So in this session we are going to discuss about the female part, also known as gynoecium. So gynoecium is the female part of the flower and it contains stigma, style and ovary. The ovary contains ovules, which is also known as Megasporangia, which is attached to a tissue inside the ovary called ovarian cavity, also called locules. So inside the ovary, the ovules are there, and the ovules are attached to the ovary with a tissue known as what? Locules. Number of ovules in an ovary can vary from one. There can be either one ovule inside one ovary, example in case of wheat and paddy, or it can be many in numbers like papaya and watermelon. Whenever we cut a watermelon or whenever we cut a papaya, we see there are many seeds inside it, right? So these seeds means what? It contains huge numbers of ovules which are being fertilized by the pollen grains inside one single ovary. Because we know fruits are what? Fruits are nothing but they are the ovaries. Next is, if one single gynoecium containing flowers are known as monocarpellary. That means if a flower contains only one single gynoecium, they are known as what? Monocarpellary. And if they contain more than one gynoecium inside one single flower, they are called what? Multicarpellary. Now in case of multicarpellary, if the gynoeciums are fused with each other, they are called syncarpous. And if they are separated from each other, they are known as what? Apocarpous. See the diagram in order to understand this thing. So we have our typical gynoecium, that is stigma, style and ovary inside it. Then we have syncarpous, which contains many gynoeciums fused with each other, and apocarpus, in which each and every gynoecium are separated from each other. After this, let us move ahead and see the ovule inside it, also known as Megasporangium. The ovule attached to the placenta, that is the ovule which is present inside the ovary, it is attached to the placenta by a substance, by a stalk-like thing that is known as funicle. The junction of the ovule and the funicle is known as what? Hilum. Each ovule has two protective layers, and these protective layers are known as what? Integuments. These integuments covers the whole ovule except a small portion is left. A small portion is kept as a pore, and that thing is known as micropyle. At the opposite of the micropyle, the side at which the micropyle is present, the opposite side of it is known as the chalazal end, and it is the basal part of the ovule. Nucellus is present within the integuments. Okay, so outside comes the integuments and then comes the nucellus, which is a food reserve, and inside this nucellus we find the female gametophyte. You can see in this diagram. So we have funicle, okay, and then we have nucellus. This nucellus, inside this nucellus we have the embryo sac, and covering this embryo sac and nucellus we have the integuments, which contains a small pore that is known as micropyle, and this is the LS of a mature ovule. So this diagram you have to draw in your exam, and along with this explanations if they ask you about the megasporangium. So note this down pretty well. Next, let us move ahead and see how this mature ovule is formed. And the formation of a mature ovule, that is a haploid ovule, from a diploid mother cell is known as megasporogenesis. So the diploid mother cell here is known as megaspore mother cell, also MMC. So this megaspore mother cell is converted to megaspore by the process of megasporogenesis. This megaspore mother cell contains a dense cytoplasm and a prominent nucleus. Okay, and it undergoes meiosis to produce 4 megaspores, which are haploid in nature. Now, out of these 4 megaspores, one megaspore becomes functional and other 3 becomes degenerate. This kind of development, where one single megaspore is developing while other 3 are getting degenerated, is known as monosporic development. Now the functional megaspore, now the nucleus inside the functional megaspore first meiotically divides to 2 nucleus, then to 4 nucleus, and then to 8 nucleus stage. Out of the 8 nucleus stage, the 2 of the nucleus stays at the middle and 3 of them reaches the 2 ends. The one at the micropylar end are 2 synergid and 1 egg cell, and the other one in the chalazal end forms the antipodal cells. So a matured gametophyte, a matured female gametophyte contains how many nucleus? It contains 8 nucleus and contains how many cells? That is 7 cells. This thing will be much more clear when you see this diagram. We have this megaspore mother cell. This one is one of the most important topic here. So note it down very clearly. This is the megaspore mother cell. This megaspore mother cell divides into 4 megaspores. Out of these 4 megaspores, 3 becomes degenerate and 1 becomes functional megaspore. This functional megaspore contains one single nucleus. This single nucleus divides and reaches the 2 ends. At the 2 ends, it again divides, and after the division, it forms 4 nucleus. These 4 nucleus again divides to form 8 nucleus, and the 8 nucleus stage looks like this. So 4 nucleus at each ends. Now 1 nucleus from each end comes to the middle, and at the middle we have 2 nucleus, and at the ends we have 3 nucleus each. So this stage is known as 3 plus 3 plus 2. This marks the formation of a fully mature female gametophyte whose center, if you see the center of the matured female gametophyte contains 2 nucleus, and these 2 nucleus are known as what? Polar nuclei. At the end, at the chalazal end, it contains 3 cells, and these cells are known as antipodal cells or antipodals. And at the micropylar end, we have 2 big cells known as synergids, and it also contains an egg cell. Out of these many cells, which cell is the most important one? This egg cell. This egg cell will be fertilized by the generative cell, which is coming from the pollen grains. Now this micropylar end contains 2 synergids. These 2 cells contains a special thickening at this end. It contains a special thick substance at the end, which is also known as filiform apparatus. This filiform apparatus helps in leading the pollen grain, which is coming from outside, to lead it towards the egg cell. So I guess this one is clear. If it is not, let me explain you once more. So the megaspore mother cell divides into 4 cells. Out of those 4 cells, 1 cell is functional. This functional cell's nucleus divides itself into 2 cells, then 4 cells, then 8 cell stage. Whenever it reaches the 8 cell stage, now see the last diagram, 2 nucleus comes in the middle known as polar nuclei and 3 at the 2 ends. The one at the chalazal end are known as antipodal cells, and the 3 on the micropylar end, out of those 3, 2 are synergid and 1 is egg cell. The synergids contains a special thickening known as filiform apparatus, which helps the pollen grains generative cell to reach easily to the egg cell. Now this marks the end of the male and the female gametophytes. Now we'll see that how these gametes are transferred. The transfer of the pollen grain from anther to stigma is known as what? Pollination. Now, depending on the source of the pollen grain, the pollination can be divided into either autogamy or geitonogamy or xenogamy. What is the meaning of the term autogamy? Autogamy means the transfer of the pollen grain from anther to stigma of the same flower. So inside the same flower, we have anther and stigma, and from the anther to stigma, the pollen grains are moving. To help this process, we need these parts to be close to each other. Sometimes the flowers expose their anthers and stigma. Such kind of flowers are known as chasmogamous flowers. Example you can see in case of hibiscus. Sometimes they are hidden, also known as clistogamous. Example viola. So in this case, the anther and the stigma are hidden. So they protect the process of autogamy inside themselves. Next is geitonogamy, the transfer of pollen grains from anther to stigma of different flower but of the same plant. Okay, so inside the same plant we have two different flowers, one containing anther, other containing stigma, and the transfer is occurring there. Genetically it is similar to autogamy, but it needs the agents of pollination. After geitonogamy comes what? Xenogamy. Xenogamy is totally different. That is the transfer of pollen grains from anther to stigma of different flowers of different plant. So you can see in this diagram, process A is autogamy, process B is geitonogamy, and process C is xenogamy. So for transferring the pollen grains from one flower to another flower of the same plant or a different plant, we need some agents. These agents can either be abiotic or biotic. Abiotic factors are wind and water. Transfer of pollen grains by wind is known as anemophily, by water is known as hydrophily. Biotic factors are known as zoophily. This pollination is very important part of the reproduction in case of flowering plants, and hence to help this process of pollination, external environment helps the flowers. So flowers in return gives them some rewards known as floral rewards like nectar or nice smell and all these things. Flowers also have a symbiotic relationship with the insects. The insects helps the flowers to pollinate, and in turn the insects can lay their seeds or lay their not seeds lay their eggs inside the flower. Generally when you if you sometimes you see that you take a fruit, you cut it, and from inside of it you see some insects inside it, right? So how does the insect goes inside it? It's very simple. When the flower is there, the insect lays their eggs. After pollination, the flower grows, and as soon as the flower grows, the insect stays inside it, and the insect starts to grow by eating up the nutrient which is present inside the fruit itself. And thus whenever you cut the fruit, you get to see the insects inside it. So they lie in a symbiotic relationship with each other. So what we have studied today, we have studied about Gynoecium, Megasporangium, Megasporogenesis, pollination, and last but not the least, the agents of pollination. We'll move further in the next lectures with these agents of pollination. Till then, see you all. Hope you all are safe. Stay safe in this situation. And thank you. Till the next session, stay safe and goodbye.